Before we start, I just want to point out I'm Serena Di C. Welcome to this webinar this morning. Here you can find my email address. And today we will talk about reagents inventory, predefined panel, staining workflows in BD Research Cloud. Okay, so I think we can get started. If you can all see my screen, if you can hear me well, if you can't, just type it in the chat. If I don't see any chat message or anything like that, I will assume that you can hear me well and you can see me well and you can see my screen. Okay, first thing first, I would like to ask you how many flow experiments you do in a year. You can type it in the chat if you want, or you can send me a specific message, private message. Okay. So what are we going to do when we do a flow experiment? Depending on how many how many experiments you do in a year, you may face several steps, right? So we first want to make sure that we have to perform an experiment. Second, we want to make sure that we do it in the right way. And then third, that we have all the necessary that we need to perform this experiment. So the first thing that we want to make sure we have is the reagents, right? So today we are going to cover the topic of the reagent inventory. So in BD Research Cloud, which is our environment for collaboration and to streamline your flow experiments, you can create or you can upload a reagent inventory. So I remember when I was back in the lab, I had my reagent inventory on an Excel spreadsheet. So I had to go from the fridge to the Excel spreadsheet to make sure that it was updated and that I had all the necessary that I needed for the experiment. So this is one of the things that we are going to talk about today, which is the reagent inventory. So I will walk you through the process of creating your own reagent inventory or just upload it from an Excel or a CSV file. Once we have done that, we want to make sure that we have all the reagents that we need. And some of the reagents may not be available on the website or on the BD catalog. So the next step would be, what if I have reagents which are not included in the website? Well, you can create your custom reagents. So we are going to cover the creation of custom reagents as well within BD Research Cloud. So just to make sure that you have everything you need in your inventory. The next step would be, which is the population that I need to characterize today? So I may be interested in working on B cells or T cells, naive B cells, plasma blasts, and so on. So how do I make sure that I have all the markers in my panel to characterize this specific population? So I will walk you through the population tab within BD Research Cloud and how this can help you to better characterize your specific population and to perform your experiment. Once we do that, we then want to create a panel, which means assign the correct fluorochromes to the correct marker and so on. So we will go through define the correct panel for your population. And finally, when we have the panel, we have the reagents, we have the population, then we need to create a staining sheet. So we need to make sure that we are actually using the correct amount of reagent for each specific marker and so on. So I will cover the generating reagent sheet part within BDRC. So first thing first, to create, to use BD Research Cloud, you can use your Flojo portal account if you have one already, which means that if you're using Flojo already. If you have a license associated to it, you can log in directly with that. Even if you don't have a license, you can have a portal account, and it's free to create for everyone. So it's important to mention that even if you want to use BDRC, you can create your free FlowJo portal account and log in with that. If you don't have a FlowJo portal account, once you go on bdresearchcloud.com, you can just click on create account. And again, this is not linked to any FlowJo license or anything like that. You can go there today and just use it right away. When you log in, you will find the homepage. So here is a list of everything that you can do in BDRC, from creating panels or finding predefined panels to create your DIVA experiments and then export that for your instrument, to then move on to workflows for your lab and storage of your data. You can load your specific cytometers that you have available in your facility or in your lab, and then, of course, creating your inventory, uploading reagents, and look at all the different populations and how to characterize them. And you can create groups within your institute or within your lab. So depending on the level of let's say, interaction that you can have an organization for your institute, or you can have an organization for your lab, and divide that into different groups for that will have each individual project. So you can parse these informations within different groups and projects. So today we will focus on the reagents and population tabs and on the panel tabs. So for reagents, as I mentioned, you can upload or import your inventory if you have already a CSV, or if you have it already on an Excel spreadsheet, and it's just one click. Or you can add reagents from the BD catalog and start creating your inventory directly in BDRC. And eventually, if you see that your reagents are not in there, you can just create your custom reagents. For each of the reagents, you can save titration information. So for example, you can define how many microliters do you need in the final volume, which is the dilution. And then, for example, in extra informations like how long do you need to stain for, the temperature, and if this is an intracellular staining or surface staining, and so on. And you can add extra notes in there. Eventually, if you need, you can, and if you have the data, you can also link specific documents for the reagents. So let's say you have done a titration, you have then concatenated your files in FlowJo, you have a nice dot plot that you want to link to these specific reagents, you can do that. And all these informations, once uploaded into your inventory, will be available during your panel design process. So you can have an information next to each reagent that will indicate if you have already that in your inventory or you don't. So sometimes we may, let's say, try to save something by using the reagents that we already have in our inventory or in our box in our fridge, and we will have this information available right away while we are designing the panel. So just imagine that you don't have to go back and forth from the fridge to your bench while you are, or to your desk while you are designing your panel, because you have everything in your hands at this point. One of the major features, I think my favorite feature is the ability to scan your reagents. So imagine today it's Wednesday, you got a package in the lab and you got your antibodies delivered and you want to add that to your inventory. If you go on bdresearchcloud.com from your phone, the only action available is the scanning. So you can scan directly the QR code of the reagent. You can, of course, zoom in, zoom out, and then by that scanning, you can add that to your inventory. You can have informations regarding the titration, for example, and you can either update the inventory entry. So let's say you've got you've got, for example, a second vial of a reagent, or you want to add that to your wish list. You can do that just by clicking on a button. What about populations? What are we going to look at in the population tab will be all the populations available. So sometimes our PI comes to the lab and says, today you're going to characterize you're going to characterize naive B cells. And so the question can be, okay, which are now the markers that allow me to characterize naive B cells? So you can go on PubMed, you do your search, scroll through the many papers that are available, or you can just go under the population tab in BD Research Cloud, look for a specific population and have the list of markers that are characterizing that population. That can be either positive or negative for that. And we will go through this as well. If you see, or if you are characterizing an unknown population, let's say you are working on something specific that you are now characterizing, you can create your custom population. What is the advantage of having populations within BD Research Cloud is for sure the ability to just click on that and have the list of markers added to your panel creation steps. So you know directly which are the markers that you need to use within your panel in order to characterize these populations. That again can be known populations or custom populations. Another tool that we have available regarding populations is the ability to look for a specific predefined panel that will allow you to characterize that specific population. So you can search for predefined, pre-validated panels to characterize, for example, T cells. You can just look for the population and you will have a list of panels that are allowing you to characterize that specific T cell population, for example. So everything is ready. You just need to do the experiment at this point. So which are these predefined panels? We have several categories of predefined panels. You will find all the OMIP, which are the published panels. So a list of all the OMIPs is available on BDRC. For each of these panels, you will find all the details. You can look at the spectral signature. In this case, this is the OMIP 102, which is the 50 colors panel. You can see at the spectral signature, you can see the similarity matrix, and you can see the hotspot matrix. Now, for those of you that are not familiar with this feature, we do have one specific webinar for the hotspot matrix in BDRC, but briefly, what allows you to do is to understand which are the hotspots that could be causing unmixing errors. So it really helps you while you are designing your panel to define a combination of markers that will not, let's say, drive unmixing errors. So it's really useful and is updated live. So you can just change the combination of markers, fluorochromes, and make sure that you are not creating any hotspot within your panel that could be problematic later on during the unmixing process. Besides the OMIP panels, we also have BD validated panels. So BD works a lot on creating panels that are that can be applied on different BD instruments. So you will find predefined panels to characterize different populations on 3 lasers, 5 lasers machines. So depending on what you have in your lab, you can choose one of these ones and eventually also use them as a base, and then just alternate or change any or replace any specific marker or fluorochrome that you don't want to use. And within these BD validated panels, we also do have the BD panel blocks, which are basically panels that have a backbone for lymphocytes. So you will have always a backbone to characterize lymphocytes, and then you can add blocks depending on if you're looking for T cells or B cells, T and B cells. So you can literally mix and match these blocks of markers, validated combinations that will help you to characterize the different populations of cells. Once we have our panel, we then need to create a staining sheet. So we would do that technically without BD Research Cloud. We would do that at the bench. We would take our calculator, we would make or our Excel spreadsheet with the formula in it. We will make our calculations and then follow the notebook where we have all the different concentrations, all the different amounts of reagents that we need to use. Well, now you can do that directly in BDRC. Once you have designed your panel, you can also split the panels into various staining steps that can be, for example, the viability step, the surface marker step for the staining or the intracellular staining. And this will be integrated with titration infos if you do have these infos within your inventory. And based on the number of samples that you want to stain, you will have the final staining volume, the number of samples, and so on. You can then export this staining sheet for records as a CSV or as a PDF. So no need of manually typing that over time. And this is just the steps that you will be prompt to. So once you design your panel, you will have all your reagents right here. Eventually, if you have them in your inventory, you can just update from the inventory, or you can go just one by one and say, I'm using the titer for this reagent 1 to 100, 1 to 200, 1 to 50, and so on. Once you have done that, you can take each individual reagent, and then depending on the final, on the number of samples and the total staining volume, you will have all your calculations right here. And then you can print out the PDF for that. So you can just bring that to the bench and start doing your experiment. So overall, you will have the full workflow without standing up. So you just can sit at your computer. You can do it from the plane if you're traveling. You can do it from your home and then get the experiment ready for the day after, and so on. So let's jump right into that. So let's demo time. This was just an introduction. And again, please, if you do have any question, ask. I'll be happy to answer your questions. Okay, so the first thing, if you have never used BD Research Cloud, the first thing that you can do, you can go on the FlowJo website. There is this BD Research Cloud tab right here, and you can sign up for free. So when you navigate to the BD Research Cloud webpage, you can find information on how to get started if you want. And then just logging in, you can either use your portal account if you have any, or you can just create a new account from here. As long as you indicate your country and your email address, you will then receive an email to sign up. But one thing that I wanted to show you before jumping into the cloud directly is the learn area and the ability to learn everything regarding BD Research Cloud. So if this is your first webinar, you will find extra informations within webinars right here. You can just navigate through introduction to BD Research Cloud. And then right here, you will find specific webinars only on BD Research Cloud, or webinars that cover both introduction to BDRC and FlowJo, introduction to FlowJo. As I mentioned before, you can store your data. And if you have ever used FlowJo, you may have seen that there is a BDRC icon on top, BD icon on top of your FlowJo window, and that's the connection to BDRC, because there is the ability to import and export your data from FlowJo to BDRC and vice versa. So all these webinars will cover part of BDRC. If you want a focused webinar only on BDRC, I recommend you to watch this intro to BDRC or to BD Research Cloud. Okay, so once we're in, this is the web page that we face. As I mentioned, there are several areas: panels, experiments, workflows, data. So today we will focus on reagents, populations, and panels. So the reagents tab, when I move on to the reagents tab, first thing I see that there are all the BD catalog reagents. I can search for any specific reagent, or I can scroll the almost 30,000 reagents available. But easily speaking, if you're trying to build your inventory, the first thing that you can do is, if you want to build it from scratch, take your box from the fridge, look for the specific reagent, and either you scan it with your phone or you just look for it in the BD catalog if it's a BD reagent. To add it to your inventory, you can just click on Add to Inventory, and it will ask you which is the group. So you once you log into BDRC, my recommendation is always to create an organization and to create a group. So you can have a group with your colleague next to you, or you can have a group for your lab, and then you can just scroll down the different groups that are there. So I could choose this one, for example. How many vials do I have of this reagent? Just one. If you have the lot information, just type that in. And then if you have a remaining volume, I know that we don't really keep track of the remaining volume when we don't have something to track it automatically. But in this case, it will be kind of easy, because if you build your inventory, you can update the amount of remaining volume time by time. As long as you use this reagent, you then will remove, like say, okay, I started from 100 microliters, I used 5, now I have 95, for example. So I can just say this is my 100 microliters, when was received, today, when expires, probably sometimes in 2026. And then which is the location. So I can say fridge 1, floor 2, which is the box, white box 1, and then the coordinates. So what is my my reagent? B6, for example. Let's see, I have something in the Q&A. Okay, I see one question, which is, what if we have confidentiality protected proprietary reagents? Can we have them on BDRC, or should we use nicknames? We can't see your reagents. Your organization is your organization. So, and you can create your own group and have your own inventory for each group. So if you don't trust your organization, let's say, or you want to keep a higher level of confidence or security, you can use a nickname as well, up to you. And here we can add some notes. Let's use it, I don't know, just Leisenbusch, whatever, and just submit. Okay, so now when the reagents are added to the inventory, you see this tab right here that says in inventory. All the rest are not in my inventory. If we don't have, if we want to add a reagent which is not in the BD catalog, we can create a custom reagent. So when we go to custom reagents, this list will be empty if you don't have any. So what you can do is either import a custom reagent or create one. When we move on to the create reagent, I can select the species. This can be my human, the target, I don't know, CD44. You can have an alternate name if you want. The hot species, the clone, the isotype, these are all the informations that you can take from the fact sheet of your reagent, and then a brand. So there are many brands in here. Depending on which is the vendor, you can select a specific vendor. You can make a new brand if you want. So I could use, I don't know, Milteni, whatever. Size 100 microliters, and then which is the conjugate. So we have 169 different conjugates from different vendors. So it's not only the ones that you find from BD, but I can find, for example, seafloors, which are not from BD, or I can find alternative names, like there are several conjugates from different vendors. Catalog number, and then eventually one note. So I can then click on add one reagent. And now I have my reagent added. In the spectrum viewer, you can also find some kind of information and use that for to add reagents to your wish list. So let's say you are designing your panel in the spectrum viewer. Here you can find different instruments. There are not only BD instruments. We have, for example, the Aurora instrument and so on. So depending on which instrument are you using, you can start and look for different reagents. And then at this point, I can take any of this reagent, add it here, and then I can add it to my BD cart right here. Add to BD cart, especially if you're working with the e-commerce, or you have the e-commerce available in your country. And then we finally end up to my inventory. So here you will find in scan reagents, you will find all the reagents that you have scanned with your phone, and you can add this to your wish list, or if you just go on scan reagent on the top right corner, you can directly scan a reagent. But I feel this is more comfortable from your phone than from a computer. I don't know, that's my idea. And then my inventory. So with the essential license, which is the one that you create for free, when you click on my inventory, you will have a pop-up that says activate a free trial. So if you want to try the inventory feature before proceeding, just activate the free trial and try it out. So here in my inventory, I can then import a CSV or an Excel sheet, or I can export one. So I think I did, let's try to export this one. That can be in CSV or Excel. So let's start exporting this one. This is my CSV. And here I have all the information from my inventory, which are which is the antigen, which is the fluorochrome, the catalog number, the alternate name, and then which is the brand, the sites, everything. So literally all the informations, how many quantities do I have, when it expires, the location, the coordinates, and so on. And I can do the same thing with importing my inventory. When you click on a reagent, here you have all the informations regarding the reagent. So eventually you can update the remaining volume. That's what I was mentioning before. If you run your experiment and you want to update the remaining volume, you can just do that. Just type the number here and then submit, and the inventory will be automatically updated, and you can build titers. So you can say, how many microliters do I need? And then which is the total staining volume, and then just click save. You will also find an activity log that says who has entered all the informations in or regarding that reagent. So when it was created, and all the different titers informations that were included in that. Okay, questions regarding the inventory? I hope this is clear. Of course, you can have everything in your wish list. And then you can update it directly from here into my inventory. If you receive a new vial, just click on the plus, or you can duplicate this one if you have a different lot number. And then here in the link, you can create a connection with your, let's say, titration information or titration plots that you generated in Flojo and so on. So everyone that access to this or that has access to this inventory can find this information. So at least we work all in the same way. We do not change things within the experiments. Okay, so once I have my inventory ready, the next step would be which are the populations that I need to characterize. So I can move on to the population tab right here. And as I mentioned before, when you move on to the populations tab, you will find different populations that are available. These are all custom populations that you see on top. But then underneath here, you will find all the literature-defined populations. When you click on any of them, you will find the publication related to that. So you can follow all the procedures that they did, and then you can find the specific markers for that population and which is the antigen density for that. So for example, negative for CD3, positive for CD19, negative for CD20, but then you have a high antigen density for CD24 and CD38 for the plasmablasts. Or if you move on to the monocytes, we know they are positive for CD14 and so on. If you want to create your custom population in BDRC, every time you want to create something, it's pretty much on the top right corner. So you can just go on create and assign a name, which is the target species, add markers for that population. So for example, CD44. Or I can add it from T cells, let's say. Okay, something like this, add markers, and then which is the expression for each of these. And then let's say CD44, add marker. So now I have my CD44 here plus all the different T cells, markers, and then I can just submit. I see there's something in the chat. Okay, which BDRC tools are free and which require the license? Just the inventory is the one that requires an extra subscription, but all the rest are free. So you can upload data, you can create panels, you can create populations, you can create experiments. What else? Everything except for the inventory, which you can still have access with the free trial. Okay, so focusing on the populations, because the next step would be our panel design. So if everything is clear for the specific populations, then we can move on to the panels. And as I was mentioning before, you can look for panels for that specific populations. So when you move into the panels tab, you will find custom panels. If you have never designed a panel in BDRC, this list will be empty, but you will find all the published panels, as I mentioned before, the OMIP panels, and the BD defined. So if I'm looking for a specific panel for a population, I can just, for example, go on the BD defined on all panels, and then I can filter by cell type. I have typed something already, so I can just look for T cells. And here I have all the predefined panels for T cells, or you can look for B cells and so on. For all the pre-made panels, you will find informations regarding the instrument. So this is 5 lasers, it's a Fortesa, and then here you can find all the different reagents. So which is the filter, the antigen, the fluorochrome, the clone, and the catalog number. If you don't have it in your inventory, you can add it. Well, if you just received it, let's say, you can add it to your inventory directly or add it to the wish list if you wish to purchase that. And then you can visualize the specific panel. Now, this is a pretty easy panel, and you can see the configuration of the specific instrument. If we go on publish panels, where we find all the OMIP panel, we can scroll down. You will see that there are all the different OMIP panels. And as I mentioned before, this is the 50 colors panel that was published maybe last year by this time. You will find the reference, the link to the citation, the configuration of the instrument, and then all the different reagents that were used. If we go under visualization, we can indeed look at the specific spectral signature, the complexity score. We can use the spectral signature graph or the combined, or we can use the stack divided by laser. Let's put it back to the spectral signature graph, okay? And then I can also hover over and see all the different filters. And then when I scroll down, I can see the similarity matrix that will indicate the similarity between dyes. And then I will see the BD spectral hotspot matrix, which will indicate the hotspot regions, which are honestly really low in this panel. But what is the advantage of using predefined panels? For sure, the fact that you can use this panel as a base. So if you want to start from something predefined and make sure that things are getting done correctly, you can start and use this panel as a base. So let's pick a smaller one, I would say. Within, for example, the BD-defined panels, these are all validated. So if you're using a BD instrument, you can scroll down, find 3 lasers, 5 lasers, a smaller panel, depending on, of course, your need. And here underneath, if you can load more, you will find all the backbones. So you will have all the different combinations here. So there will be the backbone for lymphocytes plus the specific block to characterize the B cells, or the backbone for lymphocytes plus B cells and B cell subsets, and so on. So it really depends on which is your specific target, but you will find a lot of panels in here that can be used. Let's maybe take this one. Okay, so I can go into this specific panel and I can eventually use this panel as a base. So I can add a name. I can select the group that I'm working on and a project. And then a cytometer that I have. So this was done on a Fortesa, maybe. Yep. And then I can choose to preserve the clones and preserve the antigen and fluorochrome pairing. So if I eventually want to change something, I can just remove this tab right here. And then the configuration. Okay, so then I can just click continue. And I can start my workflow from a predefined panel, a pre-made panel. So I'm just going to remove the viability die for now. And then I can select species. human. In the advanced options, I can also ask BDRC to show me only the RUO, so research use only GMP or ASR. So I'm okay with using all of them for now. And then here you will find the specific marker, then the clone. You can tag if it's an exclusion marker or not. And if you know that, you can also assign row densities. Once you are sure, I mean, this was already paired, so I can complete the step. And then all these reagents have been paired already, as you can see here. So the panel is basically done. But what I can do, just to show you, let's see, okay. If I unpair something, let's say I don't want to use that specific fluorochrom, I want to pair it with something else, I can just, for CD4, for example, I can just select it and then scroll down and see if I do have anything in my inventory. I can also tag to show me the markers in inventory only, and I don't have any for CD4. And then I can just proceed. When you start pairing something, you will see all the grayed out filters that have been already used and what could be problematic for the panel. So I could just move on something that may not be problematic as much, and I can just pair. If you see that none of these ones are good for you, still in this phase, you can actually add a custom reagent. So if you see, okay, I don't see what I have, or I don't see what I need in this list, then you can just add a custom reagent from here. So you don't need to go back and create your custom reagent. Okay, so I can just click pair. And then once all this is done, you can just complete the step. At this point, you will have suggestions on additional buffers. If you're using brilliant violet, brilliant blue, and so on, it will suggest you to use brilliant buffers. And then you have all the reagents right here for your panel. Once you complete the step, okay, you will have the list. This is eventually your shopping list. If you have something in your inventory, you will also have the option to remove that from the list. And then you can finalize the workflow. And then at this point, you can add your list to the BD cart. You can request a quote. You can add it to the shopping list. You can download this panel or email it, and then you can view it. When you view the panel, you can you see, of course, the cytometer specifically. You can visualize the panel, and then you will have the staining sheets. So at this point is where you can create a staining sheet. If you don't have the titer ratio for each marker, you can't go on, basically, because to create a staining sheet, you need to have a titer ratio, right? And since I don't have these markers in my inventory, I will just proceed by typing it in, okay? Just randomly assigning them. I will use this one, 50, just randomly typing the ratio, but you should do that correctly. This will be 1 to 100. Okay, so now I can eventually update it from the inventory if I have it, and then I can save it. I can add the staining step. So if I click on add here, I will define which is the staining step. So for example, viability staining or surface staining. So I can just take the markers that I will use for surface staining. Okay, something like that. Just typing it in. The total staining volume. And then how many samples do I have? Let's say I have 2 samples or 3 samples. And then you see that everything is automatically updated. So this can be the first step step one, and then I can create and eventually create a second step if I do have some intracellular markers or an intracellular step that I want to do. So once you are done with that, you can just, okay, save. Download the PDF. And now here is my staining sheet. Now, this is a quite easy staining sheet. I just added few reagents for the surface staining, but eventually, if you do have multiple steps, you can create multiple steps the way I showed you. And then you will have a staining sheet as the one that I showed you in the slide. Okay, I think I've showed you pretty much everything regarding panels, inventory, reagents, staining sheets. If there is any question, if there is something specific that you want to see, I'll be happy to show it to you. One more thing, going back to the question that was asked before, which are the features that come with a subscription? The sharing feature. So you can share this panel if you have a subscription, but alternatively, you can just download a PDF or a CSV and send it by email. Otherwise, if you have a subscription and your colleague is invited to the organization, you can just share it and you can share it outside of your organization. That is the same thing with data. So as long as you are collaborating internally, you can share data internally with a free license. If you want to share data externally, the subscription will help you to do that. Okay, so I think, yeah, we haven't shared anything. I think everything was covered, but if you do have any specific question or interested in seeing something specific, I'm just not covering the cytometer part, the workflows and data because are covered somewhere else. But remember that you can always access the support. So you can have here. This is showing you underneath there on the bottom left corner of the window. You can click on that page and shows you either videos or informations regarding that specific page or tab. So you can really go through a tutorial if you need to. And then you can chat within your lab or your group or your institute, and you can create different channels. So it's really meant to help collaboration a lot. Okay. So I'll stick around for a few more minutes if you do have any question. But the next thing to do for you would be sign up for a portal account if you don't have one and start playing with it. Yep. I don't see any other question or anything in the chat. And just to mention, this webinar is recorded. So in case you want to rewatch it or in case you need to go through something else, you want to share it with someone else, you can always do that. To remind you, where are all our webinars are all here under Flojo. And I think, yes, if you go under marketing material, you will always find as well some brochures for BDRC. So how to get started, an overview, the hotspot matrix, for example, all the details about the hotspot matrix and so on. So these are free to download. You don't need to sign up or do anything like that. So just feel free. Okay, so if there are no other questions, then I will give you 10 minutes back. Okay.